Hi everyone, welcome to our next video. In our first part of our chapter 8 video, we talked about the types of cellular division. In this video, we're going to specifically talk about the eukaryotic cell and how we make more cells from one cell. And this goes through a process called the cell cycle and then mitosis, which is our actual division. Mitosis is going to be its own video, so I'm going to encourage you now and encourage you again to watch that video in its entirety outside of this one, because we're not really going to get into mitosis too much in this video because there is a second. So we do have some vocab. As always, we start with our vocab so we understand what we are talking about. Our first vocab word is chromatin. This is a complex of DNA and proteins that makes eukaryotic chromosomes. The chromosomes inside of every nucleus in our body are not just DNA. They are proteins along with DNA that work together to help create that chromosome. Sister chromatids. This is one of the two identical parts of a duplicated chromosome. A centromere is a region of the duplicated chromosome that appears to pinch where the two sister chromatids are joined and where our spindle fibers will attach. The cell cycle is like the life cycle of a cell. It's an ordered sequence of events that extends the entire life of a eukaryotic cell from the time that it is created to the time that it is done dividing. Interphase. This is a period of the cell cycle when the cell is not actively dividing, and it does contribute to most of the lifespan of the cell. Mitosis is the actual division of the nucleus of the cell into two genetically identical nuclei, and cytokinesis completes the division of the cell by dividing the cytoplasm to form two genetically identical daughter cells. A chromosome structure is very important to eukaryotes. Because DNA is a double-stranded molecule that is made up of 4 different subunits that are called nucleotides. A chromosome is one long continuous thread of DNA. In human cells, as I said in our last video, have 46 chromosomes in them. We get one complete copy of our chromosomes from our mom and one complete copy of our chromosomes from our dad. So we have 23 pairs of chromosomes. Each chromosome is associated with a group of histones. These are those proteins that the DNA can wrap around. The histones allow the DNA to condense into chromosomes without supercoiling or breaking as they wind themselves up. Chromatin is the starting phase of DNA and protein before any wrapping into chromosomes begins. Before chromosomes begin to condense, we have chromatin, which is just a jumble of all 46 chromosomes and the proteins associated with them. As mitosis begins to progress, the chromosomes will condense further from that chromatin. Since DNA is duplicated in the S phase, which we'll get to in a moment, our chromosomes begin to look like an X, having both a left and a right half. They are genetically identical. And one half of a chromosome is called a chromatid, and together they form sister chromatids. So one of these is a chromatid. Here's our centromere, that region that appears pinched. This is where our spindle fibers will attach, and when we put them together into the X shape, we get a chromosome. This is our cell cycle. So we're going to go through the different phases of the cell cycle now, stopping with mitosis, which is the next video. We start in interphase. Interphase begins with a phase called gap 1, or G1. In gap 1, our cell is carrying out its normal everyday job and function. It is performing itself as a skin cell, it is growing, it's duplicating organelles, it's bringing things in and out. It's carrying about its normal functions. It must double its organelles, and it does grow a little. Again, this is that first phase of interphase. Interphase is where cells will spend the majority of their life. At the end of gap 1, before the cell can move on, the cell must pass a very important and critical checkpoint. This makes sure that the cell is ready to move into the rest of its interphase and division. If the cell is not needed to be divided at that point in time, the cell enters something called G0 or gap 0. This is where the cell will literally just do its job until it's needed to make more of itself. Then we move into the synthesis or the S phase. This is the second phase of interphase, and this is where the cell will duplicate its DNA. DNA replication is completed, start to finish, all in the synthesis phase, all inside the nucleus, while the rest of the cell is doing its everyday job. There is another critical checkpoint at the end of synthesis. This makes sure that the cell's DNA is relatively undamaged and has been completely replicated before it moves in. Our last piece of interphase is called gap 2. Gap 2 is the last phase of interphase, and it's where the cell must double in size. The cell continues to carry out its additional functions, and it prepares itself for division. The critical checkpoint at the end of gap 2 says that the cell is of adequate size. Adequate size means that it should be approximately double its original size. All of its DNA is undamaged, and all of the necessary organelles that needed to be divided and duplicated have done so. The cell cannot move into the M, mitosis division phase, until it passes this checkpoint. The synthesis checkpoint, it's just kind of like a, okay, good, DNA replication is finished. Gap 1, gap 2 are the most important interphase checkpoints. Gap 1 says, I am needed and I am ready to be divided. Gap 2 says, I am of adequate size, I have the proper nutrition, and everything is ready to go. We then enter the M phase. M phase stands for mitosis. Mitosis is the division of the nucleus and all of the contents of the cell except the cytoplasm. The end goal of mitosis is to have two genetically identical cells that are made throughout the process. And then lastly, we move into cytokinesis. And this is the division of the cytoplasm. We do have several checkpoints within the M phase, but the overall M phase checkpoint occurs right before cytokinesis happens. It ensures that the DNA has been separated to the opposite sides of the cell and that the cell is fully ready to be divided. These are the stages of mitosis. I'm not going to go through them in detail because there is another video that goes over the pictures of each phase and what they look like. But just so you are familiar, we have prophase, we have prometaphase, we have metaphase, anaphase, telophase, and then cytokinesis. Cytokinesis isn't necessarily part of mitosis, but it can start happening at the same time as telophase. And we like to refer to the stages of mitosis as PPMAT. Prophase, prometaphase, PP, metaphase, anaphase, telophase, MAT. This is a video on mitosis. I'm really going to encourage you to watch it. It is linked right into the PowerPoint. So please, please, please take a minute to watch this. It's very informative and it will really help you understand mitosis well. Cancer is defined as uncontrolled cell division, uncontrolled cell growth. We do talk about it with mitosis because cancer divides through mitosis. When a cell becomes cancerous, it has the ability to skip over all of the checkpoints in the cell cycle and just constantly be jumping right to that division phase. The definition of cancer is uncontrolled cell growth and division. And we have two different types of cancer that we specifically talk about. We have benign. Benign tumors are non-cancerous growths that still form from uncontrolled cell division. These are non-cancerous. And then we have malignant. These are caused by cancer cells that have broken away from an original tumor through a movement called metastasis. Two main treatments for cancer. We have radiation. We have chemotherapy. Radiation will kill cancer cells and help shrink the size of tumors. It is localized, meaning that it is treated just to the specific area where the cancer cells are found. And it damages the DNA of the cell so much that it can no longer divide, but it can also hurt healthy cells in that area as well. Chemotherapy is systematic, meaning that it travels throughout the whole body. It uses certain drugs to kill any cell that is actively dividing, which means that it does also kill healthy and dangerous cells, which is why people who are on chemotherapy lose their hair, their immune system weakens, their nails become brittle, because those are all cells that divide very rapidly. And again, it is systematic, so it travels through the whole body. We are going to stop here. This will be our next video, but I do encourage you to go and watch that mitosis video to help you understand the various stages of mitosis.